Genetics and inheritance. Today, guys, we'll be doing mutations, and today's date 4 April 2024, Thursday, and the time there it's quarter past one. Sorry. And for those who are new here, guys, I'll advise you to do a full screen mode and also put this on HD. So thank you guys for being here, and I would like to appreciate all my subscribers, guys. Let's move on. Today's date will all for sure most of you are going to watch this after school. So anyway, how was school today? Thursday, guys. Tomorrow it's Friday, and then then we have a short weekend, then we come back, and then we head on. Some of you will be chasing us, some of you will be chasing you. So anyhow, let's move on. And without even going anywhere, guys, let me show you this. These guys yesterday sent me this, and we did a pedigree, and previous we did pedigree, and then this was the problem faced with familiar hypercholesterol, and then it was caused by a dominant T and a recessive T, right? Recessive T. So we can come to the key, and when we check the key, it was said that those that are unshaded, they are without FH. So it was caused by a recessive TT. So we were to go and put that small letter T. You know that first step of this pedigree is that pay attention to those phenotypes that are being caused by a recessive allele, right? So with that being said, we go, and then the shaded one are there with the FH. So you know, since it's caused by a dominant allele, possibilities are two: either the person is like this, or the person, he or she, is like this. So we can come here. I was sent this by this other guy, guys. He told me that I was doing this as a what it's an activity nyana as I was studying all along. So I want to see if I was doing it right, guys. And the guy did everything right. I will show you his work, and then we can come to NOP offspring, since we know that they are the siblings, or they are the offspring of H, which is kept small letter t. It means all of them, they have small letter t, and since they have FH and it's caused by a dominant allele, it means that they are like this, they're like this, they're like this. So the G, the father, we can, how do we find this G, this father here? We can come and say these are the offspring, and he is the offspring of number S and number D. Since number D is giving small letter t, it means our G here it's small letter t, and since the other one is being with FH, it means that it's like this, right? You will see why I'm doing this only. And then he said how many, and the question was how many generations are shown in the pedigree diagram. Or they can also ask you what the diagram is this. They can also bring different questions, right? And then the family one, we are seeing it for the second time, with if not the third time, and then we can count them: one, two, three, four. Then 4 generation, right? And then number 1.4.2, where determine the number of males in a diagram above who have who are homozygous recessive for hypercholesterolemia. Then we are going to say 1, 2, 3, those that are not affected, right? 3. Then number 4 are a couple G and H. This couple here, these two couple here, couple G and H are planning to have the fourth child. Use a genetic cross to show the percentage probability of having a child without hypercholesterolemia. It means small letter t with 1%, right? So the guy went and sent me his worksheet, and as you can see from the worksheet, everything, it was so perfect and well executed. As you can see here, he also tried to, you can see that he was still trying to find out, to figure out if this is a capital letter T, since there's a possibility of this or a possibility of this. And how did he come with this small letter T? Is because this is the offspring of this and this, right? And also here you can see he showed that we were going to deal with this, because that they also asked us in the question 2.4.3, and then here the 3, and then 3 males is fine. And then here came the P1, and then said the phenotype male with FH, it was crossed with a female with yeah, it was like this. Let me see. It was a female, yeah, it is a male and a female, yes. Female with FH, and the genotype, it was fine. Then the meiosis, it was fine. Genotype gametes, it was fine. And then after that, fertilization will take place. As you can see, he even followed our first possibility first, right? Then T, then small letter T, and then I told him where is phenotype. Phenotype told me that because he was now doing it at the last minute, he forgot it. But I told him next time don't forget it. So he was going to write two of them. They are going to have they have FH, and then two of them they are going to have without FH, right? So the probability yet or 2 over 4 multiplied by 100 for that percentage, and then 50%. And everything, I told him that everything is fine. I'm also proud of you. Keep up the good work, guys. 100 out of 100. If I was marking this, my daughter. My daughter. And then the date 040. Maybe one day we'll also have to do this on someone's classwork book. So guys, that one, thank you for showing me that you are with me, guys. That was a proof, or that was a show, or that was an assurance to say, Mr. People, I'm with you, and I'm ready now to move to mutation. I can now move, because we have a weekly plan, right? Monday, Tuesday, Wednesday, Thursday, Friday. So if you are not following me, do so, guys. Follow me on my YouTube. And then we said Monday we are going to do a pedigree introduction, and then Tuesday we are going to do pedigree, Wednesday pedigree, Wednesday question and answer, and then Thursday mutation, Friday mutation or question and answer that are dealing with mutation. So after doing this, that's when he sent me to show that this guy is also moving with our pace, right, with our momentum. You know that we just agreed that every day just make up one hour aside to say I'll be doing this, I'll be doing. Some of you guys today will be writing a topic test, right? Monohybrid and dihybrid only. So you can see that we are also, our plan, it's also running smoothly. Someone told me that tomorrow we are going to write monohybrid crosses and dihybrid crosses, Mr. People. So I feel like I'm ready. I can go and test if I'm picking up, or I still need some revision. So I will do the revision when the teachers are teaching. So from there, let's find, today we'll be doing mutation, guys. Today we'll be doing mutation. There's no much to say with mutation. Why are you saying that, Mr. Mpepu? Mutation, it will be included in other questions like monohybrid, when maybe they were doing a sex linked, sex determination, blood grouping. Do you understand? So they might say mono, as you can see here, gene mutation. We have a type of gene mutation. These are the type of mutation, hemophilia or color blindness. We did about this, but we didn't say those are types of gene mutation, right? So now we are incorporating them. We are saying these are the type of gene mutation, since now we can give you an understanding behind what is mutation and everything, and what is happening with this mutation. So they might give you an example saying that hemophilia is a sex-linked disorder where a person is unable to clot the blood. Person is, and it's unable to clot, right? And then this type is this hemophilia, it's a type of a certain mutation. And then from there they can ask you what type of mutation is this. When now you'll have to know that we have gene mutation and we have chromosomal mutation. So at the end of this lesson, you should be able to define mutation, right? What is mutation? I was asking my sister earlier on, I said, hey, what is mutation? Today I'll be teaching mutation. What is mutation? It was like a gene thing, something happening with the gene. Hey, there's movies and everything. Hey, X-Men, Wolverine, and what you understand. She started to give me examples of movies that are dealing with mutation. I was like, it's fine, it's right. For when what is mutation? You, by this time, you can think of anything that can define mutation. But I also told her, mutation is 10 past 4. 10 past 4. Those who know 10 past 4, just. Why? Because mutation is a sudden change, guys. Mutation is a sudden change. This change is just happening suddenly. It's a sudden change in a genetic makeup, or you can say genetic composition, right? Let me also composition or structure, right? So they can also bring this on a one-word question, but a sudden change in a genetic structure, a sudden change in a genetic makeup, a sudden change in a genetic composition of an organism, or a sudden change in a DNA, right? So when now you know that they are talking about mutation. What is a mutation? It's a sudden change in a genetic makeup, right? So from there, they also told us that you have to know that in this sudden change of mutation, we have the effects of this mutation. This mutation can either be harmful, or it can either be harmless, or it can be useful, right? This sudden change, we are here, we'll be talking about a sudden change. At this moment, you know that we have a chromosome. At this moment, you know, or you'll agree with me that, Mr. Paper, I'll agree with you. I know that we have a chromosome, and this in chromosome we have a chromatid, and in this chromatid we have a gene, right? In this chromatid we have a gene. And what is a gene? You know that. What is a chromosome? You know that. What is a chromatin? You know that. You don't know that, you still have to go back, my guy. I don't think that you will be surviving what is coming for you if when you don't know what is gene, right? Likely enough, my videos are still there, and you can also download them and watch them anytime. So I was explaining what is this. So there's a sudden change in a genetic makeup. When we say this, okay, they will tell you that be aware that there's a difference between a mutation, and there's a difference between gene mutation and a chromosomal mutation. You should be able to differentiate these three, right? A sudden change in a genetic makeup, it can be in a gene or in a chromosome, right? So there's a difference when this sudden change is happening at a gene level. We say that it's a gene mutation. And when that is happening on a chromosomal level, on a whole structure, we say that it's a chromosomal mutation or chromosomal aberration, right? You'll see when we go to our textbook and what and learn further, dive deep into this. And then they say you should also be able to define what is gene mutation or what is chromosomal mutation. As I've said, these questions, they're just going to incorporate them during a question. So they will ask you what is a gene mutation. When now you answer a gene mutation, and then they come up with the questions that are related to gene mutation. So a gene mutation is a change in a mutation, or it's a it's a change in a genetic makeup that is happening on a genetic level, right? So at a chromosomal, it's a sudden change that is happening on a chromosomal structure or number of chromosome, right? Number of chromosome. So here's the thing. The chromosome can be chopped in half, maybe like this, during a crossing over in chiasma. You know that the chromosome has to cut itself, and then that portion, it has to be joined to the another chromosome, so that you will have a variation, right? And then another thing, you might find that something like this, or you might find that a chromosome changing the structure. That's the structure. The structure can be shortened or can be elongated, maybe like the other side halved, and then come and join to the other side, right? And then you can also find a situation where during a chromosomal non-disjunction, where we have more chromosome, that is also referred to chromosomal mutation. It's a sudden change on the genetic makeup. The makeup, it means that we only have two, but now we have more chromosome, or we have less chromosome, monosomy or trisomy 21, right? You know that. So you should be able to define gene mutation and a chromosomal mutation. By this time, you know. And then from there, they say that this mutation, the effects of mutation, it can be harmful. This mutation, this change, whether it's happening at a gene level or it's happening at a chromosomal level, it can be harmful. Let me explain gene mutation. Gene mutations like this, and then we will have a DNA like this. Then DNA is going to, when the DNA replication or DNA protein synthesis, you know that the DNA has to unwind, and then the DNA has to unzip. During that moment, that's where the DNA is being compromised for a mutation, where we can add more nucleotides, or we can remove some nucleotides, or some can be deleted, or some can be added. We have two examples of gene mutation: point mutation, frame shift. But you don't have to know that. When I just have to know that during this process where the DNA unzip, there's a more possibility, or there's more chances of the other strand, when during replication, the other strand to be added more chromosomes, or some chromosome can be deleted, right? And during DNA synthesis, protein synthesis, mRNA, when that mRNA is having that sequence of mRNA, it's having more genes, it means that the protein that is going to be produced is going to be a wrong protein or incorrect protein, or the protein that it's not fully functioning, right? So with that mutation, with this gene mutation or chromosomal mutation, it can be harmful or harmless or useful. This is what you have to know. Or some mutations can be harmful. What do we mean by harmful? Like for example, I was doing example a protein, right? What if that when that protein produces and we produce a protein that now works a different thing, maybe that protein is fighting different, it's fighting some organs on our body, it means that that mutation is going to be harmful, right? Another thing, albinism, albinism is when there is a mutation and that person cannot produce a pigment, right? So with that mutation, it's also harmful because the person won't be able to absorb vitamin D, and the sun is going to burn those people. People with albinism is also an example of mutation. And from there we have harmless mutation. Harmless mutation, we are talking about the mutation where the results are not that much harmful, or you'll see this when we do our exercise book, the harmless mutation, where that mutation is it's happening on a non-coding part or non-coding portion of a DNA, right? As at this moment, term 1, you should have learned, or your teacher should have told you that in a DNA, 98 or 99, they used to say 99% of this DNA is a non-coding DNA, and only 1% of this DNA is the coding DNA. It means that this will be our gene, right? So it means that the protein, in DNA, the only portion that is responsible for producing protein, it's only 1%, right? So if this mutation is happening on this 99 portion of the DNA, the results are going to be harmless, right? Another example, we use example of a earlobe. You find that some of us, we have ears that like this, they have this unattached thing, or some of us, we have ears like this, that they are this thing, the loop here, it's attached, right? That is a harmless mutation. And we also go and see that this mutation, it can be useful, right? It means that in harmless mutation we can have useless mutation, right? And then we can have a useful mutation. A useful mutation, it's a mutation that when it occurs to us, it can now add up to a survival of an organism. Do you understand? Like me being black, I can survive more in Africa compared to a white person who doesn't produce that melanin pigment, right? It means that if that was due to a mutation, it means that that mutation is going to be useful to me, right? And then some bacteria or the cockroaches, you know that when the cockroaches, you use doom today, tomorrow you are going to use a doom, they are no longer going to die, right? It means that their DNA makeup, it mutated to survive, or to survive under that doom, right? And then that gene is going to pass through the offspring. So it means that the new cockroaches that are going to be born, they are not going to be killed by doom. So that is the useful mutation thing, right? And then from there, they said this mutation, they can contribute to genetic variation, right? What is a genetic variation? With our understanding, Mr. Pip, genetic variation is just the difference in DNA of individual, right? You can write that down. Difference in DNA among individuals, right? So here, example here, what am I talking about here? We can use this example as we have 4 siblings. Example where you have 4 siblings, right? So in these siblings, you can find that the other one is yellow bone, the other one is tall, and the other one is short, right? These genes here, we have the sort of like same gene, since we inherited one gene from the mother and one gene from the father. And since we are relative, the word relative means that we are closer related to other people, right? So that variation, this variation here means the difference, right? It means that this difference that we are getting here, we are getting it at a genetical level. We are going to do this also when we do an evolution. You will understand this more when we do an evolution. Or no, man, the people who had these features, they survived more, and people and organisms who don't have these features, it's natural selection, right? Genetic variation, difference in a DNA. During a metaphase, you did this during metaphase random arrangement, and you also did this during random assortment and what, and crossing over. Crossing over at chiasma also bring genetic variation. It just bring a changes or a difference in a genetic. It means that I might have 99% gene from my father and 99% gene from my mother. And where now you might also have 99% gene from my father and 99% gene from the father. Because there's a variation, because there's some variation types, differences here that happen between us two, we are going to have genetically different, right? So I hope you understand that. And then there you understand that we have two types of mutations that can alter characteristics, leading to a genetic disorder, right? When we are dealing with genetic disorder, then we have gene mutation. Under gene mutation, we are going to learn hemophilia and color blindness. We still have other examples like albinism, a sickle cell, and cystic fibrosis, right? But these two, it means that they will be more concentrated too, unless they bring a sick. We did a sick cystic fibrosis, right? So with cystic fibrosis, they can also bring that this is a cystic fibrosis and it's caused by an autosomal chromosomes with still, if you still remember, chromosome number 7. And after that, they will tell you what type of gene, what type of mutation is this. When you say it's a gene mutation, forget about the point mutation and frame shift mutation, right? That's something else. They are not going to ask you about that. When the question comes, maybe they can say something was deleted on the gene. When they say something was deleted on the gene, they are referring to a gene mutation, right? So it's fine. And then after that, they're going to say, okay, hemophilia. What is hemophilia? We've learned this. Hemophilia, it's an absence of clotting factors. A person who is hemophilic, it means that when we cut that person, he's going to release more blood. We did this, right? He's going to release blood, and then the clotting factor, since it's not there, their wounds, they are not going to clot. And then color blindness, due to the absence of proteins that comprise either the red or green cones photoreceptors in the eye, right? We are going to do the human eye. You will see there's a retina, there's an human eye here, we have what, and then we have retina here, and then in this retina we have some proteins, and then that proteins, they produce cones or photoreceptors. So if when you have a mutation, and in that gene, gene or those during protein synthesis, agree, the mRNA has to come and take this message and send it to the ribosome. And when we reach the ribosome, we will have to produce a green photoreceptors. And if you find that we have a mutation, and in that mutation we can't produce that, we are going to say, when you have a disorder of color blindness, you cannot produce the protein that they are going to have to be for green photoreceptor. Photoreceptors means color. We are going to do these receptors next week, right? And then after that, we go to chromosomal mutation. Chromosomal mutation, they will deal with Down syndrome. This is an example of chromosomal mutation, right? And then they say we did this due to the copy trisomy non-disjunction during anaphase 1, right? There's unseparation of two chromosomes can lead to Down syndrome. So with this chromosomal mutation, they can, there's a karyotype, right? So they can bring type pair 1, pair 2, pair 3, pair 4, and then pair maybe 16. I saw this somewhere. On pair 16, they can bring 3 of these, and then they will ask you what type of mutation is there. Because we are dealing with a whole chromosome. I told you that a chromosome is a change, chromosomal mutation or an aberration. It's a change in the number of chromosome or the structure of a chromosome. So if it's like this, it means that one chromosome was added. This is a chromosomal mutation, right? Or they can give you a karyotype with this pair number 1 chromosome. with this pair number 1 chromosome like this. So if you can see here clearly, here you can see that this chromosome here, it has the other chromatid, but this one is shorter another chromatid. And then they will tell you that if you can see pair number 1, this is not how we expect a chromosome at pair number 1 to be like, since they will have to be homologous like this, right? They will tell you this is not what we expect our chromosome to look like. So tell us what type of mutation took place there, and then when by this time you'll know that this is what, it's a chromosomal mutation, right? And with gene mutation, they can give you maybe an example, an mRNA, mRNA maybe like this, and then they say this is A, this is U, since it's mRNA, and then here it will be A, right? And then it's going to match with another tRNA. So tRNA here, it will be uracil, adenine, and then this one will be uracil, right? This will be a T anticodon on the tRNA, right? Carrying the amino acid. So they will tell you that this maybe in a gene, in a DNA, since this one from DNA it will be thymine, adenine, and thymine, right? And they say in a DNA this thymine was replaced with guanine. Let's say for example, it means the mRNA, or in an mRNA this adenine was replaced with guanine. So either way, this is a gene mutation, right? And then when something like this, they will ask you that is the amino acid that's going to code here the same, or we are going to have a different amino acid. If we are going to have a different amino acid, it means that the protein that is going to be produced is going to be different, right? You did this during protein synthesis. By this time I know that you should be doing that. So after that, they can ask you what type of mutation took place here. You have to say gene mutation, although some can say it's a point mutation because one gene was replaced with another, right? That point mutation and frame shift mutation, it is not included on our syllabus on this, right? Although it's the right thing to say to reference to a gene mutation, it's a type of mutation, but here we're just going to hit it at the top of things, right? We're just going to say it's a gene mutation and we end up there. So we might find that we see that it's like this, it was deleted, it was deleted or it was inserted. It's a gene mutation. It's a mutation that is happening at a genetic level, right? I hope you get that. Thank you. Let's move on, guys. We can now fast go to our, can pass by our green book. We can pass by our green book. I've said a lot of things here, so we are just going to pass by. But a mutation is a sudden unexpected change in a genetic structure. You can say structure, you can say composition, you can say a makeup of a cell, right? When these unexpected changes happen, a sudden change, sudden unexpected change, it means it's suddenly and randomly. I talked about this. Mutations occur suddenly and randomly and may caused by many environmental agents such as X-ray and certain chemicals. And then this mutation can occur naturally, like a chromosomal mutation, where a chromosome is because of non-disjunction, right? That was the natural mutation. Or it can also caused by X-ray or a certain chemicals in a gene level. Maybe like, let's just take for example, the cells are undergoing protein replication, and then the complementary strand is being produced, and then the X-ray now come and cause 1G here to change structure, and then you find that G now, instead of coding with C, it's now coding with another G. So that there, it will be a mutation caused by an X-ray or some certain chemicals, right? Some chemicals can cause mutation. So but mutation may be harmful, harmless, to the organism in which they occur. Harmful mutation cause changes in a DNA that cause errors in a protein sequences that can result in a partial or non-functional protein. When we say the harmful mutation is a mutation like in a gene mutation, it's a mutation that will result in, I told you that this is what the mRNA is going to do. mRNA, let's just say it's like this mRNA, and then we say that we had a change there. Instead of A, we have AAC. Right, so it means that this the tRNA that will come and bind here, it's going to be having now uracil guanine instead of uracil adenine, right? So it means that the amino acid that is going to be carried there is going to be changed. So the protein can be produced since you know that amino acid same amino acid can be encoded by a different codon, right? So when you go and check if the new amino acid is different to what it was supposed to be produced. So if it's a new amino acid, there's a possibility of that protein to be produced, but that protein, because it has a different amino acid instead of another amino acid, that protein can be partially or completely non-functional. It means that we can just have a protein that is just moving around, hence we said it's harmful, because what if that protein now functions something different, right? Or you might find that due to this change, there is no anticodon that can come and match to this sequence. It means that that protein is not going to be produced. So we can have a no protein production, or we can have a protein production, but that protein can be non-functional or partially or completely non-functional, right? Partially, they just mean small nyan. The protein is the protein is there, but the work that this protein is supposed to do, it's limited, right? Because of amino acid thymine. They had maybe something guanine or threonine, whatever those names, 20 amino acid, right? We have 20 amino acid. So yes, with harmful amino acid we have harmless mutation, we have harmless mutation, and in harmless mutation they have no effect on the structure or functioning of the organism. We talked about this, that this harmless mutation might find that it's happening on the non-coding part of DNA. So because it's happening there, we have no effect on the structure or the functioning of an organism. So it does not play a role in the production of a protein. Or we can have a production of new things like you'll see the difference when we go to the book. We'll see the freckles and the earlobe. So if the earlobe is like that, then it's harmless. You understand? Whether you have a mutation here, earlobe, this thing being attached or not attached, it's a mutation. Yes, it's a sudden change, but it's harmless. So other that, we also have a useful mutation. They say this can be advantageous to the organism that they passed on the parents' offspring. So here's the thing, you might find that other books, they might just say we have harmful mutation and a harmless mutation, right? And then under harmless mutation we have useful mutation and useless mutation, or we have neutral mutation, or we have they can say they have neutral mutation and they have advantageous mutation, right? So same thing, useful mutation, same thing, is that mutation that when it occurs, that organism is going to benefit from that mutation, and then that gene or that sequence is going to be passed on from one generation to the next, right? But if we find that we have a mutation and this mutation is useless, it means that it cannot be passed to the next generation. Understand that this is just a knowledge based topic, right? We are learning in this topic, but when we ask you the question, we are going to just cooperate this mutation to something else, right? So maybe they will ask you, is this a useful mutation? Was that a useful mutation, or was that a harmful mutation, right? Why are you saying it's a useful mutation, right? So those type of questions. And then I also did gene mutation. These are mutations that affect a single or a few base pair in just a single gene, right? Gene mutation, it's when we are having a change at a nitrogenous base, basis, or we can say nucleotides in DNA. It's when we have a DNA like this, when these nucleotides changes, that's where we say it's a gene mutation, right? So, and then this gene mutation, it can happen at the non-coding gene, is still gene mutation, right? It's a different with a chromosomal mutation or a chromosomal aberration, but it refers to the changes in the normal structure or the numbers of chromosomes, right? I did an example and I showed you that they can say this is they say they refer to the changes in the normal structure. So it means that pair number one, we know it's like this, but if they bring you something like this, it means that the mutation happened. because we know the normal structure of that chromosome per number 1. But if it's like this, it means chromosome happen. Or the numbers of chromosome, we can have like these chromosomes, and then due to non-disjunction, you find that when this chromosome now, when this we will have cells like this, another cell without the cell, because it didn't the cell didn't separate here. And then when we have one cell or one gamete from the father coming and join here, we end up with 3 chromosomes, and this one will be the number of chromosomes being changed, Down syndrome, right? And then this one will have monosomy, and then this one will have trisomy. Trisomy just means 3, monosomy means 1, right? I don't think they'll ask you, but they can ask you that with this karyotype, right? I told you, if I'm not mistaken, I even said that there's a karyotype, where in this karyotype, right? How karyotype. I say the way I claim it. Karyotype. And then you say we have one pair number 1, pair number what. In pair number 16, they can give you 3 chromosomes, right? And they can ask you how many chromosomes are there in this karyotype. You know that all of them, all in all, they should be 46. But when we have another extra chromosome, we are going to have 47. And then they will ask you again, what type of mutation took place? You will say chromosomal mutation, right? And then they'll say give another type of mutation that you stated, or from the answer you gave at 1.2.1 example. I'm just coming up with an example. 1.1 they give you 47. 1.2 you said they ask you a question, what type of mutation is this? You say it's a chromosomal mutation. And 1.3 they can say give another example of mutation stated at 1.2. You're going to say Down syndrome. So you see, this is how they ask those questions, right? You're going to say Down syndrome. So they will give you one example. And then another thing, let's come here. Mutation results in new genotype, right? As we move from one generation to the next, this leads to the variation within species, right? The variation within species, variation iranyaka, chakalaka. When we see people in chakalaka, in variation, diversity. Yeah, diversity is not like that. Diversity means that we have impala, we have lion, we have elephants. That's the diversity. Variation, it means that we have impalas, or let's say we have elephants that are tall, we have elephants that are short, we have elephants that have a long task, long elephant track. And then you understand, that's the variation. Like us people, you might find that in a family we have something like this, right? We have siblings like this, and then in these siblings you find that the older one she's black, when as a sister you are also black, the other one ki yellow bone, the other one unali milwa washapu, you understand, the other one is very well shaped. But that's the variation. It's brought upon mutation. So an example, if you inherited one chromosome from your father and one chromosome from your mother, theoretically you should be similar, right? But you understand that there's also crossing over, there is also random assortment, and there's also mutation now that is also bringing to genetic variation. You learn that this genetic variation can be brought upon by crossing over during chiasma, and we have it can be brought upon by random assortment, and then now mutation can also bring this, right? You understand, if this chromosome is just doing its own thing without crossing over, without a random assortment. But if we had a change here in this structure, it means that that person is going to be different from another person. Ne? I hope you understand that. From there, we'll say gene mutations can cause genetic disorder. We talked about this. I said disorder. There's a difference between disorder and diseases. Go and learn it. Go and find out about this. And then gene mutation, we have hemophilia. We talked about this absence of protein needed for a formation of blood clots due to mutant gene. Here's the thing. Let's say maybe there's a gene, and then we have a nitrogenous base. When this nitrogenous base it means that the protein that was supposed to be produced, mRNA, and then mRNA produced this protein. This protein was supposed to be the clotting factor. It is not there. It means that there was a mutation and something changed in this gene level. At this gene level, something changed, and then when you can't produce the protein needed for a formation of blood clots. Do we understand? Yes. And then we also have another one, which is color blindness. Also, in this same scenario, you have a DNA, you have a gene, and then we have to have an mRNA from there, mRNA, and then mRNA will have to take it to ribosome, and then produce a protein. Because now there was a change in this gene structure, there was a change in this DNA. Let's take it like there was a change in this DNA where now your mRNA cannot produce a protein, because you understand that protein is main amino acids, right? 50 of them, 50 plus those amino acid being, do you understand? So if, like, say maybe like example, there was a change here, and then this amino acid, it was wrong, or this amino acid now the transfer mRNA, they cannot read the code there, it means that your protein can be produced like this. So you are producing a non-functioning protein. So it means that in this color blindness, you are unable to produce proteins that make up the photoreceptors in the eye. You are going to learn an eye, and then you see that in an eye we have a retina there at the back, and then that retina, when you see a color black like this from your skin, from the screen, this is photoreceptors. They are recepting photos, they are recepting these colors. You see the color, and then that thing is going to interpret. After interpreting, is going to send it to the brain, and then the brain is going to tell you that whatever you are seeing, the color that you are seeing is black. So if you don't have these proteins that are making up that photoreceptor there, you won't be able to notice that the color that we are using here is purple. You see, well, now you can tell this color is purple. So it's because that your retina there, it's not producing this. You understand? You have a mutation. And then another thing is albinism. Albinism is the absent. Also another thing, we go back to the DNA. Yes, this is hence this is a type of gene mutation. We explain this. We have the gene DNA, and then in this DNA, your mRNA that is being produced is going to produce a protein, or it's going to produce a partial or a completely non-working protein. And then this protein that is absent here, or that cannot be produced here, it forms a pigment melanin, right? A pigment called melanin, like Black Coca-Cola, the AKA song, I'm not sure. I'm going to Google it and I'm going to replay it. Hey, what Versace, what Jigama? I'm not sure it's Jigama, but there's a portion where AKA said, hey, what melanin Coca-Cola, right? So that is the pigment that is being produced. It means that your DNA as a black person, what is it that I'm proud of? I'm proud of, or the one thing that makes me to be proud is that in my DNA, I have a portion that portion produced a protein. That protein produced the melanin pigment. So that can be advantageous for me. I can absorb vitamin D from the sun. Not liqueur. Liqueur, Momakua, they are not on albinism side, because the pigment is there. The protein that is being produced is there, but they are not producing the pigment. But in albinism, hence you find that the color of albinism is not white. It's not the liqueur color. Because in a white person, the person is completely, it don't have this protein, right? So the cells that the part or the portion of a cell that produce a skin color, it's not there, right? So it's when we talk about albinism right here. So they can just give you that an albinism is an autosomal chromosomes, and then this autosomal chromosome is caused by a recessive allele, right? And then they'll say use a capital letter A or small letter a to represent someone who have albinism and someone doesn't have an albinism. Question number 1, define gene mutation. Question number 1, what type of mutation took place? And then you'll say gene mutation. 1.2, explain the type of mutation that you stated at 1.2, and then you'll say what, and then from there give 2 examples of mutation that you stated at 1.1. You'll say hemophilia, color blindness, right? But it's not only this. We also have cystic. We did this, right? Cystic fibrosis at chromosome number 7. We did this, and we also have a sickle cell anaemia. You'll see why we claim this like this. But they call it sickle cell anemia. Anemia, right? Anemia. But it's anaemia. Anaemia, right? How you claim you in this life science, you can, this is the beauty of life science in there. I can say something totally different, but if I write it down and then we find that whatever we are saying the same thing, we are right. Can come and say, hey, hi hemophilia, you see, like hemophilia, and someone, huh, what's that, what is hemophilia? And then at the end of the day, oh, he's saying hemophilia. It's an hemophilia, it's not hemophilia, it's hemophilia. But since we are Africans and we are Africanizing these names, we are going to say hemophilia, habakuku, Halloween. Okay, let's move on. And then the last one, types of mutation, we have chromosomal aberration or chromosomal mutation. Example is a Down syndrome. We did this, and when we do when we have 47 instead of 46, or when we do the chapter 2, right? So with that being said, thank you guys. Let me not waste so much of your time. You can go, this is our textbook. Our textbook, they did mutation, and then they told us that mutation is a permanent change. Here we are going, we can also pick up something that we can learn, right? Here we can learn that these change, they are permanent, guys. If we have a gene like this and then something happened here, that thing is permanent. Hence you find that today we have albinism and we cannot treat that albinism. There's no way that you can be born with albinism, but we know that it's just the problem is just that that person, he cannot produce the melanin. What if we go to the gene and insect the codon, or we insect the nucleotides that will produce that melanin, right? That's what we'll be doing when we do the genetic biotechnology next week. Biotechnology, DNA recombinant. When you saw that you have a DNA, we can come and cut this DNA, or we can come and add more nucleotides there, right? That's what we'll be doing next week. Then mutation, and then mutation can be harmless, harmful, or useful. And then we have harmless mutation, involves changes to the non-coding. Yeah, you still remember harmless non-coding, right? This DNA, it's not involved in making protein. What DNA are they talking about? This non-coding DNA. So when something is happening there, we are going to express something called harmless mutation. The mutation, the change is there. What is mutation? A mutation is a sudden change, right? Since the change is there at the DNA, it's happening, but it is referred as an harmless. Then yes, what I was saying, 99% of the DNA, but when it's happening on that 1%, that's where we can now say it's a harmful mutation, right? Then with harmless mutation, they say it does not affect the structure or functioning of a cell or organism. Where now having an earlobe, it's not going to affect you in anything. So, for example, freckles get the dot on your skin, right? You might find that there's someone more eye, what they have black dot, black brown dots, what those are the freckles. See your partners. And then we have blonde hair and baldness, whatever. Pandla. You understand? When you don't have hair, that thing is a mutation. It's a change in a gene mutation in a DNA structure, but it is not affecting you in any way, right? So they can also come a question, say, hey, a blonde hair it's an autosomal a characteristic, or the autosomal trait that is being controlled by a dominant allele or a recessive allele. And then they told you that there was a mutation that took place there. Was that a harmful or a harmless mutation? When you'll come and see what they are talking. Are they talking about freckles, blonde hair, baldness? If they're talking about this type of mutations, you'll know where not maybe earlobes or what. They think that they are not going to affect the structure or the function of an organism. That will be an harmless mutation. Then from there we'll move to harmful mutation. Harmful mutation, they change the DNA responsible for the production of a specific protein. That what I was saying, that in this, when we go to the gene side and now change that DNA, we are now going to cause the harmful mutation. Why? Because the protein that is going to be produced there, this could cause the changes to the organism physical appearance or functioning due to the incomplete or defective protein being made. I told you that we can produce a protein that is non-functional, or we can produce a protein that is partially, it can work but it's not effective, defective protein there. And then, or they can produce an incorrect protein, or it can cause an organism physical appearance like albinism. You understand the phenotype changes now. They can give you a mutation that changes a phenotype, and then they ask you what type of mutation was that an harmful or a harmless mutation. Since we are changing the phenotype of that person, we are going to say that one is a harmful mutation, right? May cause a genetic disorder, and then example will be discussed below. We discussed this previously: hemophilia, color blindness, those sickle cells, what, right? We did albinism and what. These are the type of harmful mutation. And from there, we have a useful mutation. What is a useful mutation? Also change the DNA responsible for the production of a specific protein. If the protein made increases the organism chance of survival, I talked about this, it could be seen as a useful mutation. Understand? Maybe for an example, to run faster, or to have a 5 fingers, right? It started as a mutation somewhere. Maybe we started with 1 finger or a hand without fingers, and then while the hand were producing, you'll learn this when we do chapter 9, guys, evolution, right? Then come with it, take this mutation thing, let's bring it to chapter 9, where we'll be learning the useful mutations, natural selection. But if the gene is passed on, it will lead to genetic variation. We talk about genetic variation. It will lead to DNA differences that is advantageous to a environment, to the individual. So you see, some books they will say harmless and harmful, and then we have useful and useless, or advantageous or neutral mutation, right? All of them, they are type of, then it means neutral, it will be harmless. Useful, it will be a harmful mutation, but that is harmful, that is useful, right? It's harmful, it's harming the production of protein. And then what the genetic variation is important to the process of natural selection. A natural selection organism with traits that allow them to survive in the environment are more likely to pass on to their genes. And the natural selection, this will be discussed in chapter 9, is responsible for this mutation either being passed on by the future generation. So it means that if now, like this, for example, I have here's the thing with mutation and with natural selection, it does not happen with only me passing this generation from one offspring to the next offspring. That does not just happen with only one person deciding jiggy because I have mutation I can pass it on. It also goes with time. Do you understand? Maybe like an offsprings, they are born like this, and then that when that DNA is being changed, being for an example, we have HIV virus, because you know that HIV virus is like this, and so these proteins, they undergo mitosis, they undergo mutation, so that the organism that the HIV infect cannot be, the antibodies that are going to be produced, they cannot be effective to that HIV, right? I also use an example of insects, of those cockroaches, that when those cockroaches are being sprayed by doom over and over again, they start to develop a resistant gene in their gene, or in their genetic makeup. They start to have a gene that, nah man, if I can produce this protein, it means that I can, when this doom is being sprayed to me, I can survive. And then that protein can be passed to the next cockroaches, and you find that the new generation of new cockroaches, no matter how much doom you spray to them, they are not affected. That will be type of useful mutation. So from there, we have gene mutation. We talk about this gene mutation. Gene mutation occur during replication or DNA protein synthesis. Yeah, took the protein synthesis, production of protein, because during these two processes, when the DNA it can be unwinded and unzipped, right? And you know, when you do this process, the DNA become compromised and some genes can be added or some genes can be subtracted. Here's the thing, but the a pair is added or left out or doubled up. This changes the sequence of the bases in DNA. Example of gene mutation are hemophilia, color blindness, sickle cells, and albinism. We talked about albinism, we talked about this hemophilia, absence of a clotting factor, color blindness, absence of photoreceptor proteins, right? And then sickle cells. What is a sickle cells anemia? Sickle cell anemia. They say sickle cell anemia, but sickle cell anaemia. Sickle cell anaemia. This anemia coming from the an means not, this one means blood, right? From hemoglobin, yes, hemoglobin, this is the, you can see IMO, right, to deal with blood cell. Okay, let's say what is a sickle cell anemia? Sickle cell anemia is it's an autosomal disease common in Central Africa, India, and South America. It is caused by a gene mutation which results in a faulty hemoglobin molecule being formed. If you still remember a composition of blood, I said in blood we have red blood cells, right? Red blood cells, they house something at the center called hemoglobin. This hemoglobin is responsible for carrying oxygen to the cell and CO2 to the lungs, right? You know that. So when we have this sickle cell anemia, when we have this mutation, it means that the red blood cells that are going to be produced, they are now going to be produced as like this, from the word sickle, right? Sickle kata. When we use this thing to cut grasses, right? If you still remember this, you use this thing to cut grasses, right? And then this is the sickle is coming from this, and then they produce cells, red blood cells that are moon shaped. And then they say the red blood cells which are made have a half moon shaped, something like this, hence the term sickle, right? But not only can these cells not carry enough oxygen, resulting in anemia. Anemia is a shortage of oxygen, ne? Shortage of oxygen. Where are we shorting this oxygen? Because our red blood cells cannot carry enough oxygen. Why? Because they are moon shaped. And then you say by the shape means that the cells stick to each other, blocking small capillaries. This causes damages in an organism such as brain and kidney. And then if they're like this, it means that they can now join with other one that like this, and this one can join. So you see, this joining and joining can cause the blockage in a small capillaries. When they're like this, they're just rolling, but when they're like this, they're like boomerang. Some of them they'll be like this, some of them they'll be like this. The default, right? So they will clot or they will block and the small capillary. This causes damage in organs such as the brain and the kidney. So from there, albinism, we talked about albinism, and mind you, this is just a knowledge base, a topic, right? You are learning this, and then you'll see how the question, I'll give you the activity that you will go and do as an homework for you to familiarize on how they incorporate these questions. We'll do questions tomorrow also. But albinism, it's a rare group of genetic disorder which results in a lack of the pigment melanin, right? You know that there's a DNA, this DNA, because they cannot produce the mRNA, they can produce an mRNA, but this mRNA cannot produce a protein, and this protein that can produce melanin pigment. We have albinism, but it is caused by a recessive gene mutation which prevents the normal development of a color in skin, hair, or eyes. So this is the information that they can give you and say, hey, represent D, and then represent small letter d as someone who is albinism, because it's caused by a recessive gene here. So it means, and maybe they can also do it on a family tree or a what do we call pedigree, right? And then in this pedigree, they will say those people there, they have albinism and it's caused by a recessive allele. And then they ask you what type of mutation that is, what type of mutation is this? You will say it's a gene mutation, right? And then give the specific name. It is called albinism. You get me? If they didn't tell you that albinism is what. And then they can, I also told you, I'm repeating this, give 2 examples of gene mutation. Hemophilia, sickle cell, and what, and cystic fibrosis, right? Unfortunately, unfortunately there are many prejudice toward albinos. They are often portrayed as villains in the movies, are regarded as bringing bad luck, and are at times murdered for their body parts. This one we know in Africa. They killed them. The albinos, they don't die, they vanish. They were lying. They were abducting them to kill them. Those people, they die, they are like us. Everything is like us. It's just that their skin, there's a mutation that they cannot produce that pigment. But everything, it's like us. They eat, they digest, they inhale oxygen, they exhale carbon dioxide. They, do you understand? They undergo mutation, they can go undergo copulation, fertilization, they can have babies. So everything is like, how, where is it that it's coming that albinos, they vanish and they don't die? That body, where is that body going to? So albinos have weak eyesight, and that are light sensitive and have a very light skin that is susceptible to skin cancer. They are perfectly normal in all other aspects, guys. 100%. Let's respect people with albinisms. They are just people living with that. That's what we say, that these are the people that are living with albinism. They are not disabled. No one is disabled with albinism. They are not disabled. They can do everything that we can do. Maybe their eyesight, maybe their skin, because you find that they cannot play on the sun because their skin cannot absorb that vitamin D. So the skin now bring much light to a point where it can alter the gene. Hence they say they can now skin cancer, what. They understand? Because now that light can penetrate straight to the nucleotides, it can alter the chromosomes there, and those chromosomes, maybe the chromosome is undergoing DNA replication, and then that light, because there's a race there, and then you find that that light is compromising, and it's causing another mutation that can cause the skin cancer. You will do this when you do the skin what. And then chromosomal aberration, we did this. A chromosome aberration occur during anaphase 1 if the chromosome of a bivalent do not separate, right? Non-disjunction of chromosomes. Both chromosomes go to the same pole, and thus the chromosome number of gametes changes. An example of chromosome aberration is a Down syndrome. I told you they can just give you a karyotype, and then they ask you give another example of this mutation that is taking place here. Example, Down syndrome. And then you did this in chapter 2. I'm already out of time. During anaphase 2 non-disjunction, chromosome 21 will lead to the formation of gametes. What we did this, right? And then there's the Punnett square showing that we had a non-disjunction here. This they didn't cross. This one will be anaphase 2, right? It didn't cross, and then now we have something like this, and then the gametes will have the trisomy here, and here we'll have monosomy. Mono means 1 and tri means 3. So that is from me, guys. Let me give you activity. Thank you for being here. Next week we'll be doing a biotechnology, learning how to since this DNA can change, or we can add chromosomes, or we can remove chromosomes. When we are doing that, maybe for medical purposes, you'll learn that next week, right? DNA engineering, biotechnology. So today let me give you a activity so that you'll familiarize yourself with how these questions are being asked. So this will be our activity. We are going to do this tomorrow. Homework. If I do homework, I also give homework here. Why not just be faithful to your learning and do this homework fast, maybe during school hours, because you know that when we come back from school, you will have time to rewatch this video. You can take this screenshot, write it down, take it to the school, do it during school hours, during study break, and then you come back and we do this after school, right? So thank you for tuning in. Thank you for being here, guys. If I've missed anything, we can also talk about it tomorrow. So today, that was it. This was a knowledge-based topic. Know about mutation, what is happening with mutation, 2 different types of mutation, where is it happening, how is it happening, and with that knowledge of mutation, now you can use that knowledge, or if these things can happen naturally, or if this X-ray can cause some portion of a gene to be removed. It means that we can do that purposefully. We can add some nucleotides into a specific gene to maybe increase that person's ability, right? a skin color, what you can use a baby, they understand. So we'll do that next week. Today we are done. Homework, go and do this homework. Don't forget to subscribe, don't forget to like, don't forget to share, and don't forget, guys, to refer this to your friends at school. When you have a friend, maybe a mjitanyana or a girl, you can see that this one uklachanyana li Mr. Mangmang, li Mangmang ba klachanyana, you understand. So maybe if I agree Mr. Mpepu, and then maybe he can he can give Mr. Mpepu time, and then we can do this, guys. You, in life science, there's many opportunities. You can be a doctor, you can be, to understand, here you can be a genetic counselor. Someone can come to you and say, my baby, or nah, I was suffering from what, or because my husband is tall and I'm short, I'm being scared. What if now we have a tall babies? That would, that in my family I'll just be a shortest mother. Then you might come with thing, okay, we're not in this, in your mother's side we have a those things, guys. Let's talk about this tomorrow. Today we are fine, thank you. This is the activity we'll do tomorrow, and then tomorrow I'll also bring another questions from question paper. So thank you from me, Mr. Mpepu. Good night. Thank you. Peace, guys. Stay focused, keep it 100%, keep it real. Progression over perfection. Thank you, guys. Mwah. Thank you, see you.